All right, so today we're going to be starting our topic of genetics, but before we get into that, let's go ahead and take a look back at what we were studying in the fall semester. So in the fall semester, we focused mainly on cell structure and the different biomolecules that you'll find in that cell. So this is an animal cell, a eukaryote, and you know that because it's got a nucleus and mitochondria, and we have all the biomolecules represented, like the lipids, which you will find in the membrane, primarily in the form of the phospholipids. This cell will also have carbohydrates throughout the cell, as well as proteins. Again, those are made of amino acids that are strung together, and they have those secondary structures of alpha and beta helices, alpha helices and beta sheets. And there was a fourth biomolecule that we find in the nucleus, and those biomolecules take their name from the nucleus because that's where they're found as the nucleic acids. So they came in two flavors, DNA and RNA, DNA being the double-stranded one, RNA being the single-stranded one, and both DNA and RNA are made up of monomers of nucleotides. And so just to review the nucleotide structure, we start all the nucleotides, they come in three parts. So we have first the phosphate, which is covalently linked to some kind of pentose sugar. It's going to be deoxyribose if you're in DNA and ribose if you're in RNA. And in either case, the third part of that nucleotide is going to be some kind of base. So we did mention this earlier that the nucleotides do have a direction, and that direction comes from a chemistry convention where the carbons are numbered. So we start on the innermost point here, where this would be carbon 1, carbon 2, carbon 3, carbon 4, and up in here there's an imaginary carbon. It is there, we just don't depict it, and this is going to be carbon 5. So this would be the direction of the nucleotide. It has a top side, and the top side is going to be the 5' and the bottom side is going to be that 3'. So this is considered to be the upright direction of a nucleotide, and that is going to matter for what we're talking about next, transcription, translation, as well as DNA replication. All of those are going to have a direction associated with them, right? So 5' is the top, 3' is the bottom. And so when we look at DNA structure, remember that A pairs with T and G pairs with C, and that when we're adding nucleotides, we can only add them to the 3' end of any particular strand. And again, it's always the 5' that's connecting to the 3'. So the incoming nucleotide, that 5' carbon, is connecting to the 3'. This is the only way that nucleotides can be added during any kind of polymerization of either DNA and RNA. And just a reminder that A pairs with T, so that's apples in the trees and cars in the garage, and those are hydrogen bonds that are linking those together. So that's the review of the DNA structure, right? So, hey, what's going on here? All right. So if I have a DNA structure, remember DNA is double-stranded, but these two strands are not equivalent. So on the bottom here, we have what we consider to be the correct direction, what we call the sense strand. Which is going to go from 5', because remember that's like our start, our top, and it's going to have a 3' on the other end. So the sense strand is the one that's going to go quote unquote on order. And then the opposite strand, the complementary strand, we call that the antisense strand. And so this one, remember, it's anti-parallel, so those sugars are going to be flipped, those deoxyribose are going to be flipped, and it's going to start with 3' on this side and go to 5' on the other. So the sequences are different, the directions are different. So sense strand 5' to 3', antisense strand 3' to 5'. So we are going to be talking about genes, right? And so we know that genes are encoded in our DNA. And how do we get from this DNA code to our RNA code, right? So a gene is just a heritable stretch of DNA which is going to code for some kind of protein, and that protein will either control or is the trait in the end. So for example, earlobes, whether or not they are attached or not attached, is encoded in your genes. So your gene, when it is transcribed and translated, is going to make some kind of protein that is responsible for a trait, right? So we'll go ahead and skip this part. So DNA is the master copy, so to speak. And so we know that there are two nucleic acids. DNA was one and RNA was the other. So DNA is the master copy and RNA is the nucleic acid that is going to be used to make a temporary copy of these genes during a process called transcription. So let's take a step back for a moment and talk about the central dogma of biology. So we start with DNA, which is double-stranded, and we have our A's, G's, T's, and C's. And then through a process called transcription is where we use that DNA, that master copy, to make a temporary copy called messenger RNA during transcription. And then the final part of that process is that this mRNA is then taken and used by the ribosomes to do a process called translation, where you actually translate this RNA code into amino acids, which is going to make your proteins. So these things are all linked together. That DNA codes for RNA, which codes for protein, and this is how we go from genes to ultimately traits, with that RNA, that messenger RNA, as kind of an intermediary. So let's talk first about that first step. So going from your double-stranded DNA to that temporary copy of mRNA in that process of transcription. So we have our double-stranded DNA going through this monster molecule of mysterious name and origin, which we'll talk about here in a second. So transcription. Remember, DNA is double-stranded. The strands are not equivalent. So transcription is going to occur on the antisense strand of DNA, the one that's like quote-unquote complementary backwards, the one that goes 3' to 5'. So this is what mRNA transcription is occurring on. It's not the sense strand. So be careful when you're working on those problems. If you have the option of two DNA strands, it's the antisense strand, right? Because you're going to end up with a sense strand in mRNA if you do it this direction. So transcription of mRNA happens on the antisense strand, the 3' to 5', as the template. And the enzyme, the protein that carries this out, is an enzyme called RNA polymerase. So just a reminder that every time you see an ACE, you're looking at an enzyme. And remember that the name of the enzyme has something to do with its job, right? So if I'm an RNA polymerase, chances are really good that what I make are RNA polymers. And that is in fact what this enzyme will do. So RNA polymerase will sit on that antisense strand and then make a complementary mRNA molecule that is single-stranded, and because it is using that antisense strand, when it makes its complementary mRNA, it is in the quote-unquote correct direction, the 5' to 3'. And then just a reminder that there are some differences between our DNA molecule and our RNA molecule. The nucleotides in DNA, reminder, have deoxyribose as their sugar. They have A's, G's, T's, and C's for the adenine, guanine, thymine, and cytosine, whereas RNA will use ribose for the sugar instead of deoxyribose, and instead of thymine, the RNA will have uracil. So RNA polymerase is going to come along and polymerize, make that RNA polymer using that antisense DNA template. So transcription occurs in 3 different stages. Initiation, where we start, that first nucleotide that's put down. Elongation, where we're adding some, and then finally termination, when it's saying, okay, my message is done, I can let go now. So let's talk about these steps in transcription. So we start with a double-stranded DNA molecule, and all of this DNA does not code for proteins. So DNA has a lot of different jobs. It can signal when to start. So like when you're out driving, you don't just drive on the roads all the time. There's stop signs there for you. There's green lights to tell you where to go. DNA works the same way. And the only thing that we have available to us as far as signals in the DNA is the sequence of the bases, right? Because the sugar and phosphate backbone is the same throughout. The only thing that changes is this base sequence. So some of these base sequences are going to code for protein, but some of the other ones are going to code for promoter regions. And so what the promoter region is doing is saying, hey, RNA polymerase, there's a gene here. Can you please start transcribing me? So RNA polymerase is looking around. It finds a promoter region, and then we'll sit on that area. And the first thing that I have to do if I want to use that antisense strand as a template is I have to unwind the DNA, because before it's closed off, there's no way RNA polymerase can get in there and actually start making its mRNA. So first thing it has to do is actually separate the strands so that it can get on that 3', that antisense strand, and start making base pairs. So step 1, unwind DNA at the promoter region, and now it can start its job. So this would be the initiation, right? So once it's able to do that, we can elongate. We're using, again, that antisense template strand, and we're adding nucleotides, and not just any old nucleotides. We're adding them with a complementary base. So the T will still pair with an A G will still pair with a C, but now because we're switching from DNA to RNA, instead of the A pairing with a T, the A is going to pair with a uracil. So you'll see that happening a couple times. So there is no T in RNA, so that A will pair with a uracil. So that polymerase will go through and add all of those little nucleotides in there to create an mRNA polymer. So this will continue until that RNA polymerase reaches a poly A tail, which just means it's a bunch of A's strung together saying, hey, this gene is done, you can let go now, RNA polymerase, which it does, and so that would be the termination, and then the mRNA is released, and the DNA can reanneal to itself, and we have created a separate mRNA molecule that was transcribed from that double-stranded DNA. So that would complete the process of transcription. So I just want to point out for a second, we've been talking a lot about the structures of DNA and RNA. We're about to switch to this convention of where we're only looking at the base sequence, and I just want to remind you guys where this is coming from. So this would be like an AGTC here. All of this is we're looking at the sequence of the bases from the sense strand, unless it's otherwise marked. But never forget that these bases that were attached to your nucleotide, that's where that sequence is coming from, right? And so the DNA is going to have these A's, G's, T's, and C's, and the mRNA will replace the T's with uracils. But again, never lose sight of where that base sequence is coming from. It's coming from the nucleotides. So if we look at our practice strands, okay, say we have our DNA strand, and I'm just telling you straight out that this is the 3' to 5' direction, so you could just do a straight complementary strand. So for this one, A would normally pair with T in DNA, but now it's going to be a uracil. T will still pair with A, so you would have UAA. Again, A is going to pair with U, A will pair with U, G will pair with C. So these, the old rules apply for the most part as far as base pairing. You're just going to replace any T that you would normally have paired with adenine and replace it with a uracil. But otherwise, transcription problems are going to be pretty straightforward. So let's move on to translation. So right, we have our polymerase, our RNA polymerase that separates that DNA. It's going to make your messenger mRNA and will release as you get to that poly A tail, right? So creating your single-stranded mRNA. So what happens next, right? So we go from DNA, which is transcribed into RNA, which is then translated into amino acids that are strung together in those condensation reactions to make protein, right? So we're here. Right, we have done transcription. I have my little messenger RNA molecule, and I know it's messenger RNA because of the presence of those uracils. So all of this is occurring in the nucleus. Transcription occurs in the nucleus because they're nucleic acids. That's where they found them, so that's where all this action is happening. This would be in the nucleoid region if you were talking a prokaryote. So I have my mRNA in the nucleus, and the first thing that's going to happen is it's going to exit into the cytoplasm through some nuclear pores, and it will encounter our old friend, the ribosome in the cytoplasm, right? So the ribosome is the site of protein synthesis. The ribosome is what's actually going to be doing all of the translation, right? So remember all these tiny little dots that you've been drawing since the fall, the little dots, and this was the 70S ribosomes in the prokaryotes and the 80S ribosomes when they were free in the cytosol or attached to the rough ER. So those teeny tiny little dots are all the ribosome, and the ribosomes have a really important job because they're actually using those codons, they're reading codons from the mRNA that got exported, right? So this thing that was coming here, that was coming out of the nucleus, the ribosome is what's translating that and actually making the protein in the end. And the way it's doing that is it's using these mysterious entities, tRNAs, transfer RNAs, to read these things called codons. Okay, so I'm throwing new terms at you. What is a tRNA and what is a codon, right? So let's talk about codons first. You might have noticed from some of your practice problems and in the scavenger hunt that I had you guys using triplets when we were looking at these RNA sequences. And so that's not an accident, because the triplet of the bases is something called a codon. And in biology, each codon is going to code for a specific amino acid. So like this would be codon 1, this would be codon 2, codon 3, codon 4, codon 5. And so each one is going to be associated with some sort of amino acid. So maybe that one's isoleucine. Don't look, okay, I'm making this up, guys. So it would code for some kind of amino acid. This one would code for a different amino acid. That one would go on, and so on and so on. And so remember that when you link your amino acids together, you're making some sort of polypeptide. But these codons are what is going to code, right? So it's that triplet code of what is going to equal some amino acid. And this is basically what the ribosome is doing. But it uses something called transfer RNAs to actually read these codons that you're finding in the mRNA. So, okay, what's a transfer RNA? So transfer RNA is still an RNA molecule, and remember that RNA can form those hydrogen bonds almost as well as DNA can, right? It still forms CG pairs, A's and U's will still pair up. And so transfer RNAs have these really cool, like, kind of T-shape. There are at least 64 of them. There are multiple ones that code for each of the 20 amino acids. So how these work is they carry some amino acid on one side. So like that would be up here on this one, methionine here, phenylalanine there. And where they work their magic is actually in the anticodon region, right? So it's a triplet here. So this triplet anticodon is going to match with some kind of codon. So like this CGG, if I think about, okay, C pairs with G, G pairs with C, G pairs with C, this anticodon is going to form those same hydrogen bonds that we've been looking at with a codon based on the mRNA. And so we're going to use that to really specify what my amino acid sequence is based on the codons that are here in the mRNA. So how this actually works, right? So we've got our ribosome, and we've got the mRNA that arrives in the ribosome, and it will not start just anywhere. There are special start and stop signs, just like there were for the promoter regions in transcription. A ribosome will carry along until it gets to an AUG, which is a methionine start codon. So what's going to happen first, right? So I go and I look for my AUG, and after it starts that, the tRNAs are going to start entering the ribosome, and we're looking, that tRNA is going to be looking for a match on that anticodon. So it'll come in here and say, okay, is there a match? If there's not, it goes straight out, and it will keep doing that until it finds a match. So once there's a match that's made, that amino acid will get removed from that tRNA and kind of stay there, and then new tRNAs will come in and keep looking for a match. So once a match is found, the ribosome will link those together and start to make that amino acid. So the process is always going to start with that ribosome, forming on that mRNA. First, it actually comes in two parts. So it'll form, and those tRNAs are coming in, they're looking for a match after we start with that AUG. Once the match is found, the amino acid is released. So after we have that first one, it holds on to the amino acid, and then it will just keep looking for matches. And when a match is found, it will add it to that growing polypeptide chain. So this is the ribosome that is doing this whole job here. So just to look at it another way, that we have, tRNAs coming in, and once there's a match, so this is an energy requiring process. It will add that new amino acid onto the growing chain, and then the chain will exit that ribosome. So this is how this process happens. We assemble first on the mRNA. at that start codon area. And we keep going, we keep matching until it reaches a stop signal, right? So, yeah, and then we're adding those amino acids until they exit, right? So this is just a different view of that, where I have my anticodons looking for a match, the amino acids are getting added, and we keep going our happy little ribosome until a stop codon is reached. So that's how we go from DNA to RNA to a protein product. So again, our little ribosome is coming here on our methionine. So how does it know, right? How do we get from an RNA codon to an actual amino acid? So it turns out that this has been mapped out. And so there are two different tables that you might see this in. So when we get to this triplet code, you can use this table because we're not ribosomes and we don't know how to do this on our own without our charts. And so we start with our start codon. And the codon is a triplet, so we'll have a first letter, which in this case is A, a second letter, which is U, and a third letter, which is D. So this one is going to code for methionine. Okay, that's great. How did we get there? So let's take a look at this again. So how we do this is you start, say you're starting with ACU. So let's say we start here. So my first base is A, so I'm going to come to this row of A, and my second base is C. So I come to the top here and I say, okay, where's my C? So I'm somewhere right here. And then for my third codon, I would go to U, which is threonine. So no matter what, it's threonine, right? So let's look at another example. So if it's CAC, okay, so first letter is C, I'm going to come down to row C. And then my second letter is A, so I'm going to come down to column A, so I'm right in here. And then my last letter is C. So in this case, the amino acid is going to be histidine. So that's how we interpret these charts, right? So that's how a ribosome will read it. So there are special codons, right? AUG is going to be the start. So all of these should always be start, but there's stops. So if you see a UAA or a UAG or a UGA and you're on a translation problem, do not translate past that because these are stop codons. It means that the protein is done and the ribosome would not translate past that, right? Okay. So we can skip this for now. So I do want to take a look at the other kind of chart that you'll see, which is the circle chart. If I can just click through here. Wow, this is... Oh, shit. Okay. Never mind. Clicking through. That has noise on it. Okay. So the other kind of chart that you might see is the circle chart. And so you'll get some sort of sequence like this, and first thing you're going to do is you're going to highlight whichever codon you're working on. You'll hate your life a lot less. And so how you use the circle chart, the long and short of it is you start on the inside and you work your way out. So if we're starting with, like, say U, okay, I'm going to the center in here with my U. That means that I am in this part, right? I'm in this part of the circle. After I know I'm in this part, I'm in section U, I go to my second letter, which would be A. So I'm right here in the A ring, and I'm not over here because I'm only in the U quadrant. So I'm here with the A, and then I go to my last letter, which would be C, because now I'm restricted only to the A side, and so I find that that is a tyrosine. So that's how we use that part. So, like, okay, let's say we're going to our next codon, and again, I'm just, making it easy on myself. So now my first letter is G, and so now I know I'm in my G quadrant, so I'm restricted to this part only. I go to the second line, which is my A, and then my last letter is also an A, so that is going to be glutamine. And if I do it one more time with CGC, okay, I start in the center, I'm in this quadrant here, so I go C to my next letter is G, and then my last one is, wait, where'd it go? C, so that would be arginine. So that's how you use the circle versus the table. So that's how we do transcription and translation, folks. And that's all she wrote.